So the next speaker is Ben Durham, who's currently at Rutgers Cancer Institute, and many of you know Ben has made great contributions to the genomic analysis of histio, and he's going to talk to us about RAF-Independent MEK Mutations today. Thanks, Ben.
Okay, I'd like to thank the organizers for selecting our abstract for presentation. I'll be discussing RAF-Independent MEK Mutations drive refractory histiocytic neoplasms, but respond to ERK inhibition. This will be heavily mouse model based, but also have some translational aspects. I have no relevant financial conflicts of interest, but I'll be discussing off-label uses of the ERK1/2 inhibitor ulicertinib in patients. So as we know, the systemic histiocytic neoplasms involve the monocytes, macrophages, and monocyte-derived dendritic cells infiltrating multiple tissues in multiple systems of the body. It can involve the brain, the central nervous system, the bone, the lung, and we mainly largely separated these by immunohistochemical staining. Are they Langerhans-cytosis? Do they express CD1A and Langerin, or do they not express Langerin? For many years. And we were many years like, is this even an immunological neoplasm? Is this immunological phenomenon or actual neoplastic process? And we found that, of course, it's a neoplastic process now after about 15 years of work in the molecular aspects of the histiocytoses, and about a decade of work beyond the discovery of the BRAF V600E in both Langerhans-cytosis and Erdem-Chester disease, where we see that there's an addiction to MAP kinase, PI3-AKT mTOR signaling, and receptor tyrosine kinase signaling, and more recently, non-receptor tyrosine kinase signaling, such as Syk rearrangements in juvenile granulomatosis. And that's based on a lot of work from our group and many other groups in collaboration with many of you. And we've also seen, of course, the first FDA-approved therapies, molecularly inspired therapies, be approved, at least in the United States, where vemurafenib had beautiful responses to BRAF V600 mutated histiocytoses with a very sustained response, due to progression-free survival. And then we looked at the mutational landscape. We looked at cobimetinib in BRAF V600 wild-type histiocytoses across many molecular subtypes, although we didn't see as beautiful progression-free survival when we looked into depth as we did see with the vemurafenib studies. But we did get the FDA approval for vemurafenib in 2017 for Erdem-Chester disease in adults, and then cobimetinib in 2022 for the treatment of BRAF V600 wild-type histiocytoses. But when looking more in depth into our pervasive clinical trial into what mutations were present in some of the patients who were progressing on disease, we started to understand more about different biochemical classes of the second most frequent alteration after BRAF V600 that we saw in the pie chart, MAP2K1, and more specific. And so dividing these into class 1 and class 2, and then class 3 in MEK1 mutations, where class 1 the mutation in MEK1 requires phosphorylation by upstream RAFs, such as to actually phosphorylate ERK1/2 and drive MAP kinase signaling. Class 2, you have some basal activation of phosphorylation of ERK1/2 for the mutation in MEK1, but it still requires some activation, some phosphorylation from upstream RAFs, BRAF, ARAF, RAF1, in addition to actually lead to the full activation of phospho-ERK. But then the class 3s do not require any upstream RAF involvement. The mutations in MEK1, in this case, will activate phospho-ERK fully, and are very strong activators of MAP kinase signaling, as we found. And then, more importantly, we also noticed that the class 1 and class 2s, based on work with our colleague Neil Rosen at Memorial Sloan Kettering Cancer Center, actually responded very well to allosteric MEK1/2 inhibitors, which are what's now currently approved for histiocytosis, such as cobimetinib, trametinib, binimetinib. But the class 3s did not respond well at all. And we also found this is actually one of the largest numbers of mutations and I'll show you that we found actually in our phase 2 clinical trial beyond BRAF V600E. So this leads to some knowledge gaps and various research questions. What's the frequency distribution of the different biochemical classes of MEK1 mutations across histiocytic neoplasms? How do clinical characteristics compare between the different biochemical classes of MEK1 mutated histiocytosis patients? Can MEK1 mutations drive histiocytosis development in vivo? We know BRAF V600E can, but we don't know about MEK1. What disease phenotypes emerge when MEK1 mutations are expressed at different stages of metabolic development, and early short-term, long-term stem cells, multipotent progenitors versus later in the monocyte dendritic cell-committed stages of hematopoiesis. And what are the therapeutic options for class 3 MEK1 mutations, such as MEK1 E1 or 2, I1 or 3, and frame deletion, since they don't respond very well to class 2 MEK1 mutations. So we actually, through collaboration, international collaboration over the years, we've acquired about 500 patients that we looked at by different types of non-generation sequencing, whole exome sequencing, whole transcript sequencing, showing and targeted mutation in JS, where we've seen that in this alluvio plot, the green being BRAF V600 being the most common across histiocytoses, the second most frequent being MAP2K1. But then when we look at the subtypes of MAP2K1 mutations, which include MEK1, they're class 3 MEK1 mutations. And particularly one of the class 3 MEK1 mutations, E1 or 2, I1 or 3, and frame deletion, at least in that 500 patients, was the second most frequent mutation beyond BRAF V600E. And we do see it, across histiocytoses, but it seems to be concentrated kind of in longer histiocytosis, but we do see it in Erdem-Chester disease and other non-LCH neoplasms as well. This is another way of showing that when looking at the different mutational classes, the class 3s, particularly those around E1 or 2, I1 or 3 deletion, were the most frequent alteration based on the size and the circumference of the circle of the MEK1 mutations. And when we looked at the different clinical stages of the MEK1 mutations, we see that bone was heavily more frequently involved in the class 1 and class 2, in the class 3 patients. The brain parenchyma, which is high risk in LCH at least, is more frequently involved in the class 3 MEK1 mutation patients. And then retroperitoneal and abdominal alterations, the class 1 and class 2 MEK1 mutations were more frequently involving those tissue types. And lymph node was also more frequently involved in the class 3, looking across those 500 patients. And going back to the phase 2 clinical trial with the MEK1 E1 203 deletion, where we found this mutation being very frequent and adverse in histiocytic neoplasms, we have basically the class 1 and class 2s, which had very little progression-free survival. They seemed to have a sustained response, while the class 3s did not, particularly the MEK1 E1 203 patients, did not respond, had a lot of progression of disease. And we also see that even on cobimetinib, we see a lot of progression of disease with fibrothorax, as well as the xanthomatous features of the patient's skin lesions. And also the patients who were biochemical class 2 MEK1 mutated patients who received systemic chemotherapy, 77% actually had progression of disease. So to look more into this mutation, E1 or 203, we actually developed the first MAP2K1 mutated mouse model in the world. We used CRISPR-Cas9 technology to knock in the mutation for E1 or 2, I1 or 3 deletion in exon 3 in the MEK1 mice that we created. And when we look at those after Cre recombinase, we actually see that those mice that are crossed not crossed with Cre recombinase they extract wild-type MEK1 protein with one mutation, while the MEK1 E1 or 203 mutation is present in all those that have MX1 Cre or other Cre-positive promoters crossed to the MEK1 mutant mouse. And then we're looking at the MEK1 mutation, this class 3 MEK1 mutation on these mice. We see those that are Cre-negative MEK1 mice survive beautifully, while those that are Cre positive, either MX1 Cre or VAV Cre, these are kind of both early stages or full stage of hematopoietic development, lead to a median survival of about 59 days. So they die very shortly, die within like 2 months of birth. And they develop, and I'll be showing you what kind of disease they develop. They develop a very aggressive myelomonocytic disorder that's very reminiscent, even though it's not perfectly recapitulating human histiocytosis. The mice develop anemia and thrombocytopenia. They have myelomonocytic expansion. They also have lymphocyte suppression, the B lymphocytes being the most frequent circulating white blood cell in mice. When we look at the peripheral blood of the mice, the actual cyan is actually the B lymphocytes, and this is in the Cre-negative liver controls, and it's very suppressed within the Cre-positive MEK1 mice. And then the Cre-positive also have a very large expansion of the monocytes without a larger expansion of the granulocytes. And we see suppression of the B lymphopoiesis by flow cytometry, also by immunochemical staining for B220, showing basically the loss of the white pulp of the spleen within the MX1 Cre-positive mice compared to the Cre-negative mice controls. And we see major expansion of the monocyte dendritic cell aquaphase subtypes, including classical monocytes, non-classical monocytes, monocyte-derived dendritic cells. We see the expansion of those as well, which are CD11B positive dendritic cells with suppression of the type 1 dendritic cell. And then we're looking at the tissue, both the bone marrow, EBO1 is stained for monocyte macrophage cells within mice. And we see that major expansion of this within the bone marrow, also within the spleen. We also see that the B220, again, supporting what we saw by flow cytometry, is very suppressed, is essentially negative in the MEK-1 CRE-positive mice versus the CRE-negative mice. They also develop hepatosplenomegaly. They also develop lesions within the liver that look like granulomata, which we can resolve tissues that are involved in high-grade systemic histiocytoses in humans. We also see skin lesions developing in the mice, and they develop these very early, where they develop, and their dermis develop an infiltration of EBO1 positive monocyte macrophage and monocyte-derived dendritic cells as well, with large multinucleate giant cells developing, while the CRE-negative mice do not develop this. And then we also see there's an overexpression by looking at by Western blotting. We see that the CRE-positive MEK1 mutant mice show phospho-MEK and phospho-ERK overexpression compared to their CRE-negative linear controls. We also confirm this by immunohistochemical staining with phospho-ERK1/2 in tissues, and mainly other tissues involved, commonly histiocytosis, bone marrow, spleen, liver, and skin. And then we also looked at single-cell RNA sequencing data within these mice. We looked at 3 CRE-negative mutant mice and 3 CRE-positive mice, both the bone marrow and the spleen. And these we saw promotion of myeloid skewing and expansion of mature monocytes and macrophage subsets with activation of inflammation of mutant myeloid cells. And looking on my left here, or my right, maybe your right as well. So wherever you see the gray or the white dots, those are actually related to CRE-negative MEK1 mutant mice. The red are related to MEK1 positive mice. And when you look at the other clusters, you see there's a major suppression of lymphopoiesis within the MEK1 mutant mice. You see a major expansion of neutrophil-like monocytes, classical monocytes, and non-classical monocytes, with some loss of T lymphopoiesis, but not as pronounced as in B-cell lymphopoiesis. And this is another way of showing the same thing, where we see the wild-type bone marrow and spleen, where we see that there's a major, everything basically in blue or light blue are basically B cells or B-cell precursors, and these are suppressed. And these are basically suppressed in the bone marrow with a major expansion of the monocyte subsets. And also in the spleen, we see a major suppression of B lymphopoiesis. And this core diagram is another way of showing basically relationship of the wild-type CRE-negative MEK1 mutant mice versus the CRE-positive being red, gray and red, showing those that the CRE-negative positive mice are also expressing lymphopoiesis, showing the suppression of lymphopoiesis. So basically, we also showed, looking at the top 1% differentiated cells, we saw the top 20 of those after donor cells and the neutrophil monocytes, the classical macrophage monocytes and non-classical monocytes. We saw upregulation of S100A, A8, and S100A9, which are alarmins, which cause myeloid cells recruiting other inflammatory cells into the microenvironment, upregulated in these cell types within the CRE1 and the CRE-positive MEK1 mutant mice, with suppression of the profiles supporting B-cell lymphopoiesis, also showing expansion of the chemotaxis of monocytes, and also the proliferation of monocytes is also upregulated in these different monocyte subsets, as well as ERK MAP kinase signaling, as we'd expect. So we were able to show these mice were serially transplantable. They developed basically similar disease you see in the primary mice. So basically, for neoplastic processes, you need to show they're transplantable, and we did show that with the same expansion of EBO1 positive cells that we saw in the primary mice and the hepatosplenomegaly. And then so to treat these mice, we actually set up clinical trials where we actually started treating the mice with ulixertinib versus vehicle, and the mice with the ulixertinib responded very well, and had benefit to the MX1 mutant mice over the vehicle MEK1 mutant mice, and they also had improvement of the hepatosplenomegaly. And so with the improvement we saw we also saw this improvement in mice. We actually tried to treat it in some patients that really had therapy. So we used ulixertinib in our patients we got FDA approval for individual patients, and we saw major improvement with the patients on ulixertinib after about 1 month of treatment, versus those that were not on ulixertinib, with also a major drop of cell-free DNA showing expression of the MEK1 mutant mice. So, in conclusion, the MEK1 E1 drives a lethal myelomonocytic disorder reminiscent of human systemic histiocytosis. In vivo, we see an expansion of we see that the expressing MEK1 UNO3 mutant mice at early stages of hematopoietic development and long-term, short-term symptoms multipotent progenitors have more severe disease than if you express later in hematopoietic development. There's species of lymphopoiesis and major myelomonocytic expansion. And the mice in these MEK1 mutant mice seem to have been promising actually preclinical models to study single-cell and even future combination therapies for mice. And then you start to see a promising efficacy in both monotherapy for class 3 MEK1 mutant histiocytosis patients based on our initial data and based on the mouse preclinical data and some of our single patient data. We have now started to recruit clinical trials of ERK inhibitor for patients at Memorial Sloan Kettering Cancer Center that do not respond to cobimetinib or vemurafenib. I'd like to thank you for your attention, and all my former research mentor, Omar Abdelwahab, is now a major collaborator. Of course, a clinical colleague, Eli Diamond, since I was at Memorial and just recently transitioned to Rutgers, as well as many of our other colleagues across the world, including our colleagues in Israel, in Spain, in Paris, and also within the United States. So I'll take questions now if we have time.
Thanks very much, Ben. Maybe one super quick question and super quick answer. Yeah. Gustav.
Thank you. Incredibly interesting data. My question is a practical one. How can we identify class 3 mutations in patients with ECD or histiocytic neoplasms in general? So what is the best diagnostic approach for the molecular profiling of these patients in clinical practice?
Your best diagnosis, so my question I think was finding the best diagnostic approach for locating biochemical class 3 MEK1 mutations. We actually is that in patients and following them. I think that's what I heard. So yes, we follow by next generation sequencing is the best way to find this with a large NGS panel. We do know most of from the work with Neil Rosen. We do know what the class 3 MEK mutations are. Biochemical class 3 mutations are based on a publication with Blood Cancer Cell in 2018. And we need to do NGS sequencing to follow that, to see if that mutation is present in the patients. And if they do have that mutation, then that's well known to be a class 3 mutation.
So it was an in-house array.
Yes, no. It was the MEK1 mutation that we can find the MEK. There's several types of mutations in MEK1, MAP2K1 that are known as class 3 mutations that have been established in the literature, and that we can follow that by next generation sequencing in-house. So.
Great. Thanks very much, Ben. That was fantastic.